Okay, good evening everyone. Very cold, warm welcome. It's freezing outside in Edinburgh, wherever you happen to be. Okay, this is what we've got coming up tonight. Am I getting this working? Just a few news items from me and what's coming up next. But our main talk of the evening is from Richard Shaw, who's from Edinburgh Astronomical Society, is that right? Yeah. And he's going to tell us about the search for life by someone who's done it. And then we'll have The Sky in December from Alan Pickup, which will be really good, as always. So if you're on Zoom, just a reminder to be muted. That would be great, thank you. The background I'm using for this image tonight is one that's been processed by Ian Smith. This is from one of our telescopes in Spain, and that's the Cone Nebula and Christmas Tree Cluster, which is a really beautiful image. And very seasonal as well, of course. For members, sorry to be boring, but they were due on the 1st of October, so if you haven't done it yet, please renew your subscription. And also for members, the AC 100 survey, our centenary survey, if you haven't done it yet, please complete it. The details from the newsletters, and it's the last chance, and we will base our planning for the event on the numbers that we have already. So you don't want to miss out on that. So if you haven't completed it, please do it as soon as you can. Thank you. Lots of ways of staying in touch with us through our website, where I'll tell you everything that's going on in society, through a Facebook page, X or Twitter, for now anyway. Lots of videos on YouTube of all our meetings over the past few years. And we have lots of amazing pictures from our members on several Flickr groups. We're also on Instagram now and the Blue Sky social media channel. This is what we've got coming up over the next few months. So next Wednesday we have our monthly imaging and observing group. That's for members only. On 15th of December we have an online only talk about the science of the Palomar Observatory. That should be really interesting. And then first one in 2024, on the 5th of January, we have Dr. Alex Shaw from the Royal Observatory at Edinburgh telling us about the Euclid Space Telescope, which is very topical at the moment. That's a hybrid meeting, so we'll be here at the Augustine United Church, and we'll be on Zoom for members and on YouTube for visitors. On the 19th of January, the History of Innovations in Telescopes. That will be online only. 2nd of February, another hybrid meeting, listening to Einstein's Universe, the Dawn, an exciting future of gravitational wave astronomy from Professor Martin Hendry. And on the 16th of February, another online-only meeting, the second part of Mary McIntyre's History of Women in Astronomy. If you heard the first part, you know it was a really good talk, and Mary gave us another excellent talk last month as well, which is really good. And this is the last time I'll mention it, but if you're followers on Eventbrite, then this is probably the last talk that will be on there, because we don't want to pay the exorbitant fees that they are now charging us. So just follow us on our website, Facebook, X, Twitter, or Blue Sky, and visitors will always be welcome to our talks. But that's really it from me. So it's over to Richard Shaw for our talk. So just be a couple of minutes while we sort this out.
Good evening, everybody. Hopefully you can hear me okay. Well, unfortunately that's not shown up on the screen. Let's try the long way. That's the way. You and... Right. First of all, I've got a small idiosyncrasy that some of you may have encountered if you've ever heard me speak on any topic. I've got a minor speech impediment, I've got a stutter, and my mother was a qualified speech therapist, and she said, if you're going to be speaking to an audience, then sook hard sweets. So it's rude to sook hard sweets and not pass them round. So I always bring sweets to these events, and have as many as you like. I don't need to take them home. My children have long since left home, and they therefore won't steal it all. My own background, to explain why I'm someone who's searched for life. I spent 40 years in the NHS doing microbiology, and that qualifies me to talk a wee bit about microbiology, and most of what I'm going to be talking about is indeed microbiology. But the interesting thing about astrobiology, it's a fascinating science. It's in the position right now that cosmology was 100 years ago. It's a science with no evidence, because nobody knew anything about how the universe started. 100 years ago, there were quite a few guesses. Obviously, most of them were wrong. And astrobiology were in a similar position. The only place in the universe we know about that has life is here on Earth. So a question for everybody, because I like to do audience participation. What's the oldest living creature on Earth? Anybody know? Actual creature, not species. Right? Sorry? I don't think they're. I think it's fossilised now. No, you're all guessing badly. That bristlecone pine to be over 5,000 years old. That actual tree, and is believed to be the oldest living creature. What's the largest living creature? Does anybody know? It's a mushroom. Right, this particular example of this particular mushroom is believed to be about 2,500 years old, but it might be as old as 8,000 years old, and it occupies about 3.4 square miles. It's immense, obviously. You can, believe it or not, you can eat these things, but they're mildly poisonous, so you need to cook them really well before you do. And the Norwegian Food Safety Organisation is saying, no, you shouldn't really eat them. So when we're talking about life, we start off in the unfortunate position of having a bias about it, because we think life is what we recognise as life, but it's not necessarily. It can be other things. So when I asked this question in my house of my family, they said, largest living creature, it's a whale. Oh, sorry, it's not. Oldest living creature, it's a tree. It's a redwood tree. No, it's not, but it is a tree, so you're half right. So, sorry for extraterrestrial intelligence, is that it's a waste of time because we don't even know what we're looking for. And we're spending a lot of money on what? I don't know. Because we don't even know what intelligent is. Right? Yeah. Now, we classify life as, we classify life by comparing the similarities and the differences. So, for instance, does anybody know how to tell the difference between a butterfly and a moth? Because you get brightly coloured moths and dull coloured butterflies. Does anybody know how to tell the difference? Actually, no, they both got antennae. True, that's true. But it's something more obvious than that. I've never looked that closely, to be honest. It might be true, but the really obvious sign is that when they're not flying, butterflies fold their wings over their bodies and moths behind their backs. That's how my daughter expressed it. Butterflies do that, moths do that. Right? Can you tell the difference between a rabbit and a hare? Partly, but... Sorry? They do have longer ears, yes. They also don't live in burrows, they live on the ground. And they run, they don't hop like rabbits do. They're actually very easy to tell apart when you've got one beside the other. Can anybody tell the difference between a buffalo and a bison? Everybody knows you can't wash the dishes in a buffalo. So it's about the similarities and the differences are just as important as the similarities. Now, the life we are mostly familiar with are plants and animals and fungi, which fall into a big group called eukaryotes. And the thing about eukaryotes is they have different cell structures and completely different. They have internal cell structures that are completely different from the other life forms we're used to. The one I'm going to be talking about most today is bacteria, because that's the one I know most about. And it's actually a completely fascinating subject, almost as fascinating as astronomy, but not quite, but a wee bit more dangerous. And a group that wasn't known to exist until the mid-1980s and wasn't popularly accepted until the mid-1990s, called the archaea, which preceded bacteria. The interesting thing, though, about archaea is that once upon a time, all life on Earth was archaea, and Earth had no oxygen in its atmosphere, and it probably looked a lot like Mars. And if you had tried to live on Earth, you wouldn't survive because there wasn't any oxygen. And in fact, the reason why there is oxygen is because archaea eventually developed the ability to photosynthesize, a side effect of which was they produced oxygen, and that oxygen was released into the atmosphere, and that changed everything. I'm not going to go into the biochemistry why it changed everything, because that's not that interesting. So, the characteristics of life. Right, how do you define life? There's no accepted definition of life, but NASA's got one. And NASA's definition is it's a self-replicating chemical system that can undergo Darwinian evolution. Now, I don't like that definition because it sort of goes back on itself. It's life because it's life. And I'm not comfortable with that. But what are the characteristics of life? Well, living things, and you're one yourself, they respond to their external environment. So everybody probably recognised it was cold today. You've all wrapped up to get here, and you're all going to wrap up again to get home. Organisms grow and change. Once upon a time I was that size, and now I'm this size. And I'm a wee bit old and grey. And some of you will get to that stage in your own lives, if you haven't already. You metabolise and respire, because you breathe in and breathe out, and you eat food and turn it into grey hair and fingernails and whatever else. And you maintain an internal environment, and of course reproduction. And in that process of reproduction, you pass traits onto offspring. Okay? Now, as a definition of life, that's not bad. The last one that goes along with that is structure. But it's not a bad definition of life, but it poses an immediate question. We're still suffering from the effects of a pandemic, which I'm sure you noticed, which was caused by a virus. So are viruses living creatures? Now, this is a particular virus called T-even bacteriophage, and it's got a head and a tail and legs. And the interesting thing about that is it's a virus that infects bacteria. It doesn't infect animals or plants at all, but infects bacteria. And the way it operates is it attaches with the wee leg things onto bacterial cells, and that causes the elongated body, and you can see a screw shape in that one up there, not quite so much in there, but it causes it to twist like that, and it injects its genetic material into the bacteria. And what the bacteria then do is they start making virus. They stop making bacteria, and eventually the bacterial cell fills up with viruses, and then it bursts, and then all the virus particles come out and go and infect other bacteria, which is pretty well what viruses do. So viruses don't do much. Do they respond to an external environment? Well, they do in a very limited way. Come to that. Do they grow and change? Nope. They don't. They get assembled. They make the cell they've invaded. Assemble them. Do they metabolize and respire? Nope. Do they maintain an internal environment? Nope. Do they reproduce? I suppose you could say that they do. Do they pass traits on to offspring? Yes, they certainly do. Do they have a structure? Yes, they certainly do. Now, do they qualify though as living organisms? And for instance, I'm going to be doing some numbers later on, and I'm starting now. In a litre of seawater, there are normally about 10 to the power 10 virus particles of various kinds in a litre of seawater, right? In every litre of seawater. How much seawater is there? If you want to have a wee guess, please do. But basically, they respond to the external environment, but only to the extent that they attach to something. And in the case of this virus, it causes the change that makes the DNA get injected into the cell that it's invading. there's no growth or change in that process. They don't metabolize and respire, and they don't maintain an internal environment. And reproduction depends on your definition of reproduction to some extent, but they do pass traits on to offspring and structure. So I'm going to operate on the basis that viruses are not living creatures. They are, in fact. And an interesting thing about that is these wee spacey bits, there and there and there. These are called spores, and these spores are extremely resistant to external factors like chemicals and drying, and I'll come back to them later. Whoop. Never pick them up off the floor. So, what lessons can we learn for that? I need to get rid of that, don't I? Yes, I do that. Cool. Yeah. So, it's very hard to define life, so I'm going to try and define life by defining death. What sterilization means is not the physical removal of organisms, right? So if you have a suspension of bacteria and you boil them, the bacteria are still there, but they're dead because their proteins have been denatured. You can filter things which would remove the organisms. So you can sterilize things that way, but they're still capable even the dried organisms are capable of metabolism, and that's where the endospores are important, because they lie about for very long periods doing nothing at all. And then if you put them in a nutrient environment, they germinate and they turn into bacterial cells and they start doing what the bacteria do. In the case of Clostridium tetani, they produce tetanus toxin, which is one of the deadliest toxins known to man. You don't see much tetanus about nowadays, but the commonest way to get tetanus in the UK, believe it or not, is dog bites. And the commonest way to get tetanus in New York is human bites. And a fairly common way to get it in the UK is to using animal fertilizers in the garden and stick rose thorns into your fingers, which might contain the spores, because the spores will survive for long periods, even in dried fertilizer. So the spores are actually extremely good at maintaining an internal environment. And they have a structure, and they have the means to turn into the other structure that allows them to do the other things, the reproduction and metabolizing and all that kind of thing. So how do we define it? Well, death is defined as the permanent loss of the ability to reproduce, because you can stop the bacteria from reproducing, but they might still be metabolizing. And it's the permanent loss of the ability to reproduce. Now, you can permanently lose the ability to reproduce if you're a human. It doesn't mean you're dead yet, because this is at a cellular level. It's not the entire organism. So a wee bit of detail about some bacteria, just to make it interesting. Most bacteria are defined as being gram-positive or gram-negative, and gram-positive, whether they're gram-positive or gram-negative, is because of a staining technique called gram staining, and Gram was a Danish bacteriologist in the 1870s or 1890s. When I first started working in the laboratory, folk were going, just gram that, and I'm going, surely you can't be bacteria, they're tiny. But nobody was trying. It's a staining technique. And gram-positive bacteria retain gram stain, and gram-negative bacteria, the stain gets removed quite easily. It's mainly to do with the structure of the cell wall, but I won't go into detail. In terms of shape, you get spheres, which are referred to as cocci, and this particular one is called staphylococci. And staphylococci comes from the Greek word staphylos, which means a bunch of grapes, because they look a wee bit like a bunch of grapes. You can also get streptococci, which appear in chains, and some that just live on their own or in pairs. Rods, rather, gram-negative rods in this case, they're referred to as bacilli. Some of the older bacteriology texts refer to all bacteria as bacilli, but they shouldn't. There are also curved rods that we refer to as vibrios, and spiral ones that we call spirochetes. And here's a good example of it. This is an organism called Borrelia burgdorferi, which causes Lyme disease. That's those squiggly things there. The big red blotches are red blood cells, and the great big thing with a black centre as a white blood cell, which gives you some idea of the scale as well. So spiral organisms are known as spirochetes. Have we ever found life on other celestial bodies, though? And the answer to that is yes, we have, believe it or not. The shock news. This is Alan Bean on Apollo 12 in December 1969. Now, the most memorable shops in those days, so I could see some of what was going on in Apollo 12, and all I could see was this sign at the bottom that said, TV camera isn't working. Water or some other solvent. Water's the best solvent as far as we can tell, but we've never seen life anywhere on any other planet apart from this. And all of the ones on Earth require water. An energy source. The energy source could be the local star, in our case the sun, or it can be chemical. It's believed that most of the it was believed by some that the original life on Earth started in the underwater as an energy source. I think possibly the most interesting thing about life on Earth, though, is that life started on Earth maybe 500 million years after Earth was cool enough, which says to me that it must be a relatively easy thing to do if you're able to do it that quickly. You also need some kind of substrate, and the substrate is a wee bit of a loose word there, because the substrate doesn't need to be solid, but basically they need somewhere to live. And in bacterial terms, that's usually somewhere that's either water itself or a suspension. The actual elements, the chemical elements of life, the principal one's carbon. We're a carbon-based life form. It may be possible to have silicon-based life forms, but carbon's just much more flexible, easier to do. You need hydrogen, most abundant element in the universe. There's plenty of that about. You don't need, strictly speaking, oxygen, and in fact oxygen is very toxic to some bacteria, including the archaea, but it certainly speeds up metabolism immensely, so it's useful. Nitrogen's very handy because all proteins require nitrogen, and phosphorus is handy because nucleic acids require phosphorus, and sulfur, and actually several others. Now, it's nothing complicated about that, but it tells us something. Because life can't have existed until these elements were present. And these elements were created inside stars as a process of nuclear fusion. Population 3 stars have almost none of these elements. So if you're a population 3 star and you've got a planet going around you, there is no life there because there's no chemicals to make that life. Population 2 stars are poor in what astronomers call metals, which is everything that isn't hydrogen or helium. So basically, there had to be nuclear fusion going on long enough and supernovae exploding long enough for these chemicals to be distributed about before life could form. Even having said that, though, the universe is about 13.8 billion years old. That's about 4.5 billion years old. So possibly the reason why we can't find life on any other planet is we're first. Isn't that a scary thought? And I've just told you that bit. So this is actually where life and astronomy come together, because you can't have life until astronomy's done the things it needed to do. The universe had to come into existence, and then the stars needed to start fusion, and then supernovae needed to blow up and spread it about. So my view of astrobiology is that what we're actually looking at is things that can survive a hostile environment. And a hostile environment can be lots of different things. For example, high temperatures. What's the definition of a high temperature? There will be no life on the sun because basically there aren't even atoms in the sun, it's plasma. The electrons get blasted off by the very high levels of energy. Dry conditions is a hostile environment. The bristlecone pine I told you at the beginning survives in what are referred to as arid conditions, but it still requires water, so there's not no water. And actually, dry conditions, those of you that wash dishes in the traditional manner without using a dishwasher, dishwashers are excellent because they are very good at sterilising your dishes, as well as washing them, because of the heat. But the important thing, if you're just washing them by hand, is not to wash them, it's to dry them. Because most bacteria that are dangerous will not survive drying or try to survive on dishes that have been dried. Now that assumes you've washed them properly because they can live inside any food that's still stuck there because it'll absorb water to the atmosphere. But drying your dishes is what makes them safe for using the next time. Competition from other bacteria, and I've specified bacteria here, that's because I've said nature's full of examples of bacteria surviving in hostile environments. Acidic environments, I'll come to some examples of these later, are important as well. And the human body, right? As far as bacteria are concerned, the human body is a hostile environment. They find it hard to survive there. It's a fight for them, although they have some advantages. And indeed, the whole universe is a hostile environment. I read a book once, which might have been written by Carl Sagan when I was much younger, and it said that the universe is not hostile, it's just indifferent. It doesn't care whether you survive or not, because it's the universe and you're only you. Why would it worry? And I believe that. I believe that to be true. So let's go through these conditions. This is an organism called Alterchiales I'll need to read that again. Hamiconexum. It's a very complex organism. It's a strict anaerobe. Oxygen kills it extremely quickly. But what it does is the cells connect to each other through these wee appendages. And that's a big example of an appendage. And they communicate with each other that way. Now remember, these are effectively the size of bacteria. They're about a micron in size, about 1,000th of a millimetre. But they're very different from bacteria. Where they live is in deep. The interesting thing about it is it produces the aforementioned endospores, and you can see them quite clearly, that one there and that there. And that allows them to survive in very dry conditions. Now, Bacillus anthracis has been isolated from Egyptian mummies that have been mummified and left underground for 5,000 years. The spores survive, the bacterial cells themselves don't, but the spores do, and they've been recovered. They've been brought back to life, 5,000-year-old bacteria, and they just happily carry on as if nothing had ever happened. Just a wee interesting side story. If you ever work in the health service, you'll meet interesting side stories. I was, for reasons that I won't bother describing, moved to the Southern General once upon a time in my career, just at the time when they were dealing with an outbreak of anthrax in the south side of Glasgow. Now, the source of that anthrax was apparently, and yeah, okay, no problem. And they said, well, fly it over to Prestwick Airport, because we've got a facility there. We'll fly it over and we'll bring it in and we'll treat your patients. But it's expensive. It's $28,000 a pop. You mean each dose? Yes, $28,000. Right, that's fine, no problem. It's your money, it's your drugs, you do what you like. So they came along and they started giving it to these people who recovered or didn't, and then they were discharged. And then, being intravenous drug abusers, they do what intravenous drug abusers do. They disappeared. They went underground. And the Americans go, you don't get a hold of these people? No, of course not. What are you doing? What are you expecting? But anyway, that's just a wee side story. So Bacillus anthracis, love this organism simply because it's actually quite nasty, but it does interesting things. And here's another one. It doesn't infect humans. It's called Delovibrio bacteriovirus. And that's it. That there and that wiggly bit. That's another cell. And what it actually does is it comes along and encounters a bacterial cell and it basically eats it, eats the inside of it, punches through the cell membrane and eats the contents of the cell. Which I find bizarre, but it's true. That's actually what it does. One that can cause human infection, Helicobacter pylori. I don't know if any of you have ever had Helicobacter. It's actually very common in the human gut, but unfortunately causes disease sometimes. My father, when I was much younger, he had surgery for a stomach ulcer. And a few years later, about 3 years later, I found out that his stomach ulcer was probably not needing surgery. What it needed was them to treat the Helicobacter pylori that was causing it. The Helicobacter pylori was very controversial as a cause of disease at the time. It was a couple of Australian doctors who said, don't bother with surgery. What to do instead, don't bother with these fancy drugs that you're developing at huge cost. What you want to do is use some relatively cheap antibiotics and stop this organism multiplying in the stomach. And everybody went, especially the big American drug companies who had a lot of money invested in it. And what they discovered was that the world didn't want that radical change. So they decided to do something radical themselves, and what they did was they infected themselves with Helicobacter pylori, got stomach ulcers, treated it with erythromycin and got better, just like that. Now the environment it lives in is inside your stomach, and your stomach has got stomach acid, and please don't try this at home, but stomach acid is strong enough to dissolve nails. Literally it will dissolve a 6-inch nail. As I say, do not under any circumstances try that at home. What Helicobacter pylori does is it produces an enzyme called urease, and it produces lots of it. And this enzyme breaks down urea, which there always is some of in the stomach, into ammonia, and that microenvironment neutralizes the hydrochloric acid. So if you could wash away the ammonia somehow, Helicobacter pylori would die relatively quickly, but they just create this microenvironment and survive very readily. So it's a good example of surviving a hostile environment. I'll have a wee look at numbers now because there's quite a lot of bacteria. Bear in mind that smaller creatures tend to exist in much larger numbers than big creatures. Right? There are a lot more blades of grass than there are trees, for instance. And there are on Earth just slightly more than 8 billion humans right now. I had to update this not long ago because the number's gone up. Every single one of us carries a biome, and your biome is basically the bacteria you carry in and around you, well, inside you mainly, but on the surface of your skin, coating your teeth, in your ears, and up your nose. And that biome for each of us consists of somewhere between 10 and 20 trillion bacteria. And if you do a wee bit of simple arithmetic, that's 10 to the 23 bacteria in or around the human population of this planet. Right? And if you're going, so what? Remember that, depending on your estimate, there's only 10 to the 11 stars in the galaxy. So there are 10 to the 12 times as many bacteria in the human population as there are stars in this galaxy. And that, I have to say, I find a wee bit astounding. So, bacteria are found everywhere, and even underground in the subterranean biosphere. And they've been discovered up to 2.5 kilometres underground, and they might be further down than that, but nobody's got there yet. How they got 2.5 kilometres underground, I don't really know, but they do. And you find them down in deep mines, and if you drill for them. They're also found in hydrothermal vents underwater, where the temperature is about 121 degrees centigrade. And there are just an awful lot of them. It is estimated that the total number of bacteria on the planet is somewhere between 1.5 and 2.3 times 10 to the 27. Now, if you're going 10 to 27, I don't know what that means. But I'll give you a clue. See if you take 10 to the 27 and you half it. 10 to 27 is a huge big number. Half it's still a big number, because half it is 5 times 10 to the 26. Right? You actually need to start dividing up by huge amounts before you have any effect on that 10 to 27. More on that topic in a minute. This is an example of one of those underground bacteria. It's called the Geogemma barosii. It was found in a South African gold mine about a kilometre down. Its energy source is sulphur, and oxygen is extremely toxic to it. The temperature at the bottom of that mine is about 120 degrees centigrade. If you've ever been down a mine, you'll know that they get warmer the further down you go, for reasons that I personally don't completely understand. So if I haven't scared or fascinated you already, I'm going to try again to fascinate you. There are an awful lot of different kinds of bacteria. In the normal human gut, there are at least 500 different types of bacteria. And we have successfully isolated about 300 of them, and most of the rest we don't even have names for them. Right? There's a couple of hundred types of bacteria in there that we don't know anything about, apart from the fact that they seem to exist, which is itself just a wee bit scary. And that's you. You're carrying that about with you right now. You've all heard of domestos. What does it do? It kills 99.9% of bacteria. Now, do any of you put domestos down your drain? Good. What a waste of time and money. Because as soon as you do, they're just going to multiply again. If I apply that 99.9% of bacteria to the total human biome, if you were to get all the people on the planet and mash them up so that the bacteria get let loose and drop them into a huge big vat of domestos, it would kill 99.9% of them, and it would only leave 10 to 20 bacteria, and incidentally, no people. So that's the kind of number that makes you go, ugh. But again, don't try this at home. So how do we, if we're going to go out to the planets, if we're going to go to Mars, for instance, how are we going to find microbial life there without actually bringing samples back? And the methods available to find organisms are these ones. There's a direct methods where you basically look down a microscope. It's not a very good way of finding bacteria because you need to point it in the right direction. It's a bit like saying, I'll get a telescope and find an exoplanet, because you might find an exoplanet, but it's actually quite hard. It's harder to do than it sounds, and you need other techniques to go with it. The best one currently in use is culture, because what you do there is you get bacterial cells and you allow it to multiply, and then they become visible and you can see them, then you can separate one from another very readily, which is mostly what bacteriology. The other one is looking for metabolic products, like the results of the metabolism and respiration. And here's an example. It's not the wee boats, it's that. That's a metabolic product of bacteria. It's gone on for rather a long time. I don't think we've found any limestone cliffs anywhere on Mars yet, and I suspect we won't. That takes us back to the Viking landers. 1976, I think the Viking landers were, correct me if I'm wrong. The Viking landers were the first attempt to find life on Mars. There was two of them, not surprisingly called Viking 1 and Viking 2. And they had 4 experiments on them, and the 4 experiments are a pyrolytic release experiment, which is basically you heat something up in the absence of oxygen and like evaporate the whatever it is you're testing. And it was completely negative, didn't find anything. There was a gas exchange experiment where you're basically trying to find the respiration of an organism, principally oxygen being turned into carbon dioxide, that's the main one, or hydrogen being turned into water. Nothing happened there either. A labelled release experiment, which is where you would use radioactive carbon or radioactive hydrogen as a source, and then you would hope that life would take that up and you would be able to find the radioactive material within it. Not a lot happened there either. And the last one was the GCMS, that stands for gas chromatography mass spectrometry experiment, and that wasn't a great success either. So one of the 4 of them, and I can't even remember which, is sometimes reported as having given equivocal results, but none of them found anything that convinced them that there was life on Mars. And of course, all the things we've been doing since, we've sent many more explorers since. We haven't found anything that we can definitively call life, apart from a meteorite that actually landed on Earth. However, the GCMS experiment wasn't a complete waste of time, because in most microbiology laboratories, certainly all the ones in Glasgow and most of them in Scotland now, you find one of these machines. It's about yay high, and it looks for all the world like a drinks dispenser without the window and the place to put the coin. And what you do is you make a suspension of bacteria on a slide and you dry it, and you stick it in that wee slot. And to summarise what the machine does, it points a laser at the slide and it evaporates the bacteria off the surface of the slide, and then it effectively sniffs it. And you can use that. There's a fingerprinting method, I suppose, where you can identify bacteria very quickly and relatively cheaply if you can afford one of those machines, which themselves cost an arm and a leg. I've just put that up again for no other reason than to say metabolic products are dead interesting, but what can we do about it? If we have, though, a plan in their heads that we're going to isolate bacteria growing in Martian soil or any other planet to that matter. Most of the soil organisms that you find on Earth don't grow very well in a laboratory environment, don't grow at all in a laboratory environment. And one way of doing it is this thing called an I-chip, or an isolation chip. And in effect what you do is you get the bacteria in very small numbers, and you put them in this and you basically try to grow them in soil. That diagram explains it a wee bit better than I can. One bacteria was placed in each pod, and you bury it in soil, you leave it for a while, and you hope the bacteria will grow, because that's their natural environment. But trying to grow them artificially generally doesn't work. So there are lots of bacteria growing in the soil about which we know not very much, because if you can't get large numbers of them, you can't actually study them very readily. So I'm not quite finished, because I'm going to show you a wee video, and I like this video. If there are any of you who do not believe in Darwinian evolution, look away now, because I'm going to show you that it happens. Because the point about evolution in bacteria is it happens fast enough that you can see it over the course of, in this case, I think it was about 10 days, because they multiply at a huge rate. E. coli, for instance, which is one of the commonest organisms studied, partly because it's very easy to study and very easy to grow. If you put it in an ideal nutritional environment, it will double its number every 20 minutes. Which, if you're any good at doing logarithms, it will tell you that by the end of the day, if you put it in that environment, the mass of bacteria would be greater than the mass of the Earth. So, I'll interrupt this, incidentally.
So what we ended up building was basically a petri dish, except that it's 2 by 4.
Right, that's just a slab of nutrient medium. That's, I think, 4 by 2 metres. It's been divided up into sections. And each section, the outer sections, there is no antibacterial substance, so the bacteria will grow freely. In the second section, because it's coming from both sides, there is a level of antibiotic that's expected to stop bacterial growth. And from that, and survive on Mars, then they will have survived on Mars. But they might be 2.5 kilometres down. They might be able to survive with the perchlorates. They might only live on the top of the mountains. They might only live where there's lots of water and carbon dioxide as ice in the poles, and we just haven't looked there yet. And that's the challenge that we have. But I've got another problem with that, because, and I've got 2 of them. One of them is this one, a spirochete called Leptospira hemorrhagiae. And the reason I like that one is because it's dead hard to spell, but I can spell it. Ha-ha. The Leptospira hemorrhagiae causes a disease called Weil's disease. It's transmitted by rat urine. It's quite common in, well, insofar that happens, it's common in sewage workers, because they encounter quite a lot of rat urine. It's just an interesting organism. The fun thing about Leptospira, though, this is one of the organisms that we cannot grow in artificial culture. And obviously from the size of it, you could probably tell that's an electron micrograph. Because in a microscope it doesn't look quite as interesting. And the key thing about it is the wee hook at the end. It's curly, but there's a wee hook at the end. Right? A wee hook there, and a wee hook there, and a wee hook there. The other one is called Erysipelathrix ruseopathiae. And I like that one because I can say it as well as spell it. And Erysipelathrix ruseopathiae is taught in medical microbiology as being difficult to differentiate from particularly Listeria monocytogenes. You'll have heard of Listeria, not necessarily the monocytogenes, but it's supposedly difficult to differentiate. Well, that's actually nonsense. It's dead easy to differentiate the two. First of all, Erysipelathrix ruseopathiae kills pigeons and not guinea pigs, whereas Listeria kills guinea pigs and not pigeons. But we long ago gave up injecting animals with bacteria, partly because it's expensive to keep the animals and partly because it's just cruel, and there's better ways of doing it. But there are some biochemical tests you can do that can differentiate them easily. And anyway, they don't look even remotely similar down a microscope. But the real reason why I like this one is because in the laboratory where I worked, there was a group of people that I won't identify who were considered to be worth paying more than the rest of us. And they didn't really know anything, to be absolutely honest. They learned their bacteriology from reading Commando comics, I suspect. But I did it properly and I knew what these organisms were. And One of them said to me one day, could you have a look at this? And that's actually what I saw down the microscope. Could you have a look at this and tell me what it is? And I happened to spot the clinical history on the request form. And the request form said, shepherd with skin infection. And it causes a disease called erysipelas, which is a skin infection that you get in your hands if you deal with sheep. Right, so right away I'm going, I know what this is, without even looking. And as soon as I looked at it, I said, it's erysipelas ruseopathy. And she said, it's what? Erysipelas ruseopathy. It kills pigeons. And everybody else in the lab was going, it kills pigeons. Did you not know that? Which made me feel a wee bit superior for a moment. But anyway, that's my favourite organisms. Have you any questions? Please ask.
Okay. Oh, thank you, Richard. That was really fascinating. Any questions about pigeon killing? I don't know. So we'll start with questions in the hall. If you're on Zoom, put your questions in the chat. And if you're on YouTube, also put your questions in the chat. We'll hopefully get to you now. So questions here. One from Fran.
Hi, that was really, really interesting.
Thank you.
A bit scary, because what you didn't mention was the whole idea of bringing something back from Mars to us. And we might be a positive environment, but that wasn't my question. My question was, what's the longest surviving spore that's been discovered?
The longest surviving ones would be ones that have been rescued from Egyptian mummification sites.
Right.
Where they're about, I think the oldest ones are probably about 7,000 years old.
And still viable?
They are still viable. Not necessarily infectious organisms, because there are plenty of organisms that aren't infectious, but certainly still viable. And they're relatively easy to get back to life. You just put them in used to an environment and give them a day. Following morning, there they are. Hello, where have you been?
Okay, thank you.
Hi. You're concerned about the potential of us taking something to Mars. What about, I mean, they're obviously amazing, amazingly, what's the word? You know, that takes a lot to kill them off. Yeah?
Robust.
Yeah, robust, thank you. But what about just the spacecraft? So the unmanned spacecraft that are going. I know that they're very careful, the environments that they build these things in. But supposing there was something on the surface, is it possible that it could survive the journey?
It could, but the numbers is what makes a difference.
Right.
The number of bacteria surviving on a spacecraft that's sent to Mars, bear in mind it has to survive the journey and all the radiation that's there, is the number to start with is likely to be low in the maybe hundreds, possibly thousands, but for bacteria that's a very low number. And they die off in a logarithmic fashion. So 90% of them will die quickly, and of the remaining 10%, 90% of them will die not quite so quickly. But it doesn't take long before you get to the stage where you're at no surviving organisms. Humans, on the other hand, are carrying about...
Variety of these things.
Well, yes, but there are bacteria. There's an organism called Streptococcus radiodurans, which will survive on top of a nuclear pile. It's just radiation resistant, utterly radiation resistant. It doesn't seem to be affected by the sort of thing that would make you glow in the dark from a distance. If there's one such organism, there might be another. And obviously that organism, by the process of Darwinian metabolism, came from something. And I don't know what the evolutionary advantage is of being able to survive enormous levels of radiation, because actually radiation is one of the main drivers of evolution, because that's what causes mutations to happen in the nuclear material. So I'm not really sure what the evolutionary advantage is, but if you're going to live in a nuclear pile, you might as well be resistant to it.
Questions here? Okay, have we got questions on Zoom? Oh, so we've got one here. You're good.
Given all that, are there any techniques we can use to successfully find what we could maybe call life on other planets?
Yes and no. The first problem is, what do we mean when we say life? Right? And forget intelligence, because I don't think we even know what we're looking for in intelligent life. But we're not even sure what life would be. It's not completely beyond the realms of possibility that there's something that's replicating and metabolizing and reproducing and passing on traits to their offspring and all the things that are put in as defining characteristics. But we would see it and not recognise it. Maybe because it does so very slowly over very, very long time periods, or maybe because it does it very quickly over extremely short time periods, or maybe because it does it in ways that you would consider to be science fiction. And I've read science fiction that says there are things that live in the sun and stuff like that, and I go, not very likely. But one thing I've learned in doing astronomy and microbiology, nothing's impossible. And that's the only thing I can say is nothing's impossible. There is no ready means, though, of finding life on other planets unless we're going to say life like us that we would understand. I think a point to make there is that even bacteria are much more similar to us than we think they are. Each of your cells, for instance, has within them something called mitochondria. Mitochondria are the bits inside cells that produce energy. And it's widely believed that these actually originated inside eukaryotic cells as bacteria being absorbed by cells and just saying, well, don't do anything else except produce energy. And they're going, oh, fine. And that's a symbiotic process that came about 4 billion years or 3 billion years ago. Who knows how long. And that's not a very clear answer. I know I'm giving you a very, very vague answer, but that's as much as I can tell you.
Just don't know what NASA is spending all their money on then.
Well, as I said at the start, if you're spending money on the search for extraterrestrial intelligence, then just get to me instead and I'll waste it in much more interesting ways. I'm much more likely to be successful.
Okay, no more questions in the room. Was there anything on Zoom? Have we anyone?
Andy Thomas has raised his hand.
Okay.
Yeah, hi, it's Andy. What's the mass of bio as opposed to the mass of the rest of us? And do we have an estimate of the mass of bacteria as opposed to, say, mammals and insects on planet Earth?
The short answer is I don't know. But I did say earlier that 90% of the dried weight human feces is bacteria. If you're going to talk about mass, it depends on what you're actually measuring, and that's why the dried weight is important in that respect. But what I will say is, my understanding is that the mass of insects hugely outweighs the mass of humans. And I also know that all of the human population could stand on the Isle of Wight, although why they would want to, I'm not sure. But that's as much detail as I can give to answer that question, and it's not very much, sorry.
That's okay, thanks very much.
Any other Zoom questions? Nope. Will, anything on YouTube? No, not on YouTube. Okay. Well, can we thank Richard again? It was a really fascinating talk. Slightly icky in places, and thanks for the sweeties. Thank you.
You're welcome.
Okay. So now we're going to have the sky in December, always a good month for astronomy, and Alan Pickup's going to... Sorry. Alan Pickup's going to tell us about that. Okay. That's fine. Thank you. Skip.
Can you hear me, Houston? Right, I think we're go. Let me bring up my laser pointer. Oops. And that's not working. I'll see if I can go on without it. Okay, right. Sky in December. As Mark said, it's an interesting month. Usually is in December. For one thing, we have the best meteor shower of the year. But there's a number of other things of interest happening this month. One of them is happening tonight, and we have an auroral display. I don't know whether you've been looking at our WhatsApp group, but Ian Smith has posted a nice picture of aurora that he's caught. So if it's clear later, then there's a chance that we'll see an aurora. There was about 2 or 3 solar flares in the last few days that have disturbed the Earth's magnetic field, so we're getting an aurora. For the ephemeris for the sun, this shows, let's come up, yes, the rise and set times of the sun during the month into next year, and the twilight times. We take the nautical twilight times as being effectively when it's dark enough to see the stars. So the nautical twilight times are changing, not very much during the course of the month, because the sun's turning a corner, as it were, at its most southerly point. In fact, the sun is further south at the winter solstice on the 22nd of December, the small hours of the morning. If you look at the rise and set times of the sun, you'll notice that the latest rise time of the sun occurs after the solstice, and the earliest set time of the sun in the evening occurs before the solstice, and that's because the Earth's orbit is elliptical, and the sun is ahead or behind this, so it's not exactly due south at midday, at 12 o'clock precisely. Also that we're, of course, west of Greenwich. So besides the sun, the moon, these are the phases of the moon through into January. You'll see the moon is currently between full moon and last quarter. So it's a gibbous moon, still quite bright. New moon's on December the 12th, and the period around there, a couple of weeks around there, is the dark of the moon. So we see our darkest skies, and we have the darkest and longest nights of the year. The full moon on the 27th of December, sometimes called the cold moon, or the long night moon, or even for some reason the oak moon. So I don't normally go into moon names, but that's the full moon names for December. Excuse me. The planets for the month. This is a sort of banner, a letterbox shape showing the entire 360 degrees of the equatorial sky stretched around from left to right. You'll see the sun is down here. This is the bit that's in daylight. Oh, you can't see my laser. The sun is in daylight, obviously, but the rest of the area is in darkness. That curvy yellow line we call the ecliptic, and that's the apparent track of the sun around the sky being highest in the summer when the sun is above Orion in the sky, and it's at its lowest. Now, in fact, this month, it's moving during December from the constellation, was spending most of the month, in fact, in the constellation of Ophiuchus, which you won't find in any zodiac. Only on the 18th of December does it move onwards into Sagittarius, which you will find in the zodiac. As for the planets, well, we won't see Mars this month. It was in conjunction with the Sun on November the 18th, and it's too deep in our morning twilight to be seen this month. Of the planets we can see, well, the brightest by far is Venus, which is lagging the sun in the sky. A brilliant morning object. Some of us watched a daylight occultation of Venus by the moon on the morning of November the 9th. I don't have any images of that to show you, but that was visible. I watched it quite nicely through more than binoculars. The other inner planet is Mercury, which is just about barely visible this month. At the moment, in fact, until the middle of the month rather, it's hugging the southwestern horizon in the evenings. And then it goes through inferior conjunction on the sun's near side on the 22nd of December. And at the very end of the month, last 2 mornings of the month, it just becomes visible in the morning twilight, in the brightening morning twilight. but it's going to be better seen in January, and I won't mention it anymore tonight. In the evening sky, we have Saturn moving from the south at around nightfall to set in the west-southwest for Edinburgh at 22:33 tonight, and progressively earlier each night. And not far from Saturn, above and, I can't point it out with a laser, but above and to the left of Saturn on the diagram there, let's flash it, is the planet Neptune, which is one of the 4 planets visible in the sky tonight until Venus rises. The brightest planet we have is Jupiter, which is conspicuous in the evening sky now. It was at opposition on November the 3rd, just less than a month ago, and it's due south 47 degrees high in the south at 21:51 GMT tonight. And it sets, as it moves the plough below the pole in the north at these times, beginning to climb in the northeast as the constellation of Cassiopeia, the W of Cassiopeia, passes overhead. The summer triangle is sinking in the western sky, still there, even though it's not summer anymore, as we know to our cost. And we have a region opposite the summer triangle of Orion and Taurus, which are climbing in the southeast. The 2 bright planets I've already mentioned, Saturn down in the southwest, and Jupiter more or less in the south at these times. And of course, the main feature of December sky is the Geminids meteor shower, which has its radiant point near the star Castor in Gemini and is climbing in the east at these times. Normally, I start these talks with a view looking north, so I'm going to break the habit of a lifetime by beginning our tour of the sky by looking to the west rather than north. The summer triangle I've already mentioned. It's dipping down in the western sky. Deneb, Vega, and Altair. You'll see them in the lower half of the center of this chart. And it means that this is, by this time, interesting features in the summer triangle are getting a bit low for observation. But it is rather higher earlier in the night as it gets dark. Deneb, for example, is 75 degrees high in the southwest, as seen from Edinburgh, at the end of nautical twilight tonight. Oh, that's better. Pegasus is, you'll see, dipping lower into the southwest. And what I want to look at is how bright it is. It's been outbursting, it's been changing in brightness about magnitude 9 early this week, and it was this comet was discovered back in 1812, and it's got an orbital period of 71 years, so it's making its 3rd return since it was discovered. Now at perihelion, its closest point to the Sun is 117 million kilometers from the Sun, which is just outside the orbit of Venus. And it'll get there next April the 21st. Tonight, though, it's 388 million kilometers from the Earth and out beyond the orbit of Mars. It should brighten because it's moving inwards, and it almost certainly will become a naked eye object before it dips into our western evening twilight in March. Now, it's been dubbed the Devil Comet, one area of interest, the north of the summer triangle. In fact, this area of interest, because on the image of the comet, that the coma has striations in it like horns, and it's gas and dust streaming away from the nucleus. As I say, there appears to have been another outburst of the comet in the last couple of days, and I picked this view of the brightness rise of the comet since 3:24 GMT on November the 30th. The observer himself was in Romania, but I'm not sure where the observations were taken from, possibly there. You can see it's brightened considerably in this over this just a period of less than 2 hours. It's brightened from around 12.75 to almost magnitude 10.3. So that's a brightening of the center region of the comet as a new outburst occurs. This is the view to the south in the mid-evening. As I say, all 4 of the planets are visible. The furthest west of them is Saturn. By this time, it's getting a little low. And that's the details for Saturn for the middle of the month, magnitude 0.9. It is fading slightly as it recedes. The diameter of the disk is 16 arcseconds, and the rings span 37 arcseconds. They're tilted towards us. The north face is tilted towards us at 10 degrees, which is 5 degrees more than it was, less than it was a year ago. So those rings are gradually closing up. It's getting lower in the southwest, in the southern sky, or in the southeast, towards the southwestern sky, and it's setting earlier in the west-southwest each evening. Jupiter is the brightest object in this area. There it is. Jupiter, a good deal brighter than Saturn, and it's to the south of the star Hammel in Aries, the main star in Aries. And it's obvious as it climbs through the east and the southeast each evening, and it's passing high in the south just after these times, in other words, bit after 21 hours tonight, while we're still in the meeting. Details, well, there they are. Magnitude minus 2.7, starting to fade, and 46 arcseconds across, so it's quite easy to see and to photograph the belts of Jupiter and the 4 main Galilean moons. This is an image taken by David Brett of the ASE, and it does match up quite nicely with this computer-generated image in the middle that I produced for December the 16th. That image by David Brett was taken on November the 8th. The other 2 planets I've mentioned are on show. One of them is the distant planet Neptune, which lies to the east of Saturn and north of Saturn, and it's just south of the circlet of Pisces, the loop of stars that represents one of the 2 fish of Pisces. Neptune is magnitude 7.9. So you'll just about see it through binoculars in a dark sky. Telescopically, it's only 2.2 arcseconds across, and its distance is 4.47 billion kilometers at this time on December 16th. On the other distant planet, a bit nearer, is Uranus, which lies between, as I say, Jupiter and the Pleiades, and it's rather brighter at magnitude 5.6, and it's 3.7 arcseconds across, and at a distance of 2.81 billion kilometers. Now then, turning our attention to the constellations, the region south, the image is the winding constellation of Eridanus, the river. Saturn itself is in Aquarius, the water bearer, and although you can't really see it, but just below and to the right of Saturn in the early evening, we have the constellation of Capricornus, the sea goat. So you can see all the constellations there have got a watery theme. The constellation further south of Saturn is Piscis Australis, which is a southern fish, and that's also watery. So that area has got earned the name of the Celestial Sea or the Celestial Ocean. Now, for this view, with the Stellarium planetarium software, I've turned on the feature that allows you to show the brighter, messier objects, and you can see some of them. Again, the fact that I can't point them out with a laser is annoying. The 2 yellowish objects on the right-hand side, labeled M15 and M2, are globular clusters. They are above and to the west of Saturn on this picture. The top one is M15, and there it is, imaged by Mark Phillips. It lies 35,700 light years away, and it's one of the oldest globular clusters, about 12 billion years old, magnitude 6.2. Now, to the south of that, or below that rather, on this image, is M2, and that's slightly fainter, magnitude 6.5, and it lies in the constellation of Aquarius. And again, I'm showing Mark's image. It's further away than M15. It's something like 55,000 light years away. And I said M15 was old. Well, this is just as old, something like 12 billion years old. I can highlight a region here which I want to focus on, taking the square of Pegasus and Andromeda. There it is, blown up. Because I have to mention, because they're quite high in the south in our evening sky this month. M31, the Andromeda galaxy. To get to M31, you take a line along the top of the square of Pegasus, and you extend it, and then make a right, and go out to the region of the oval smudge of Messier 31. And here is an image of that taken by Pat Devine. Of the ASE, wonderful spiral galaxy, not very dissimilar in size to our own Milky Way, magnitude 3.4 and 2.5 million light years away, and getting closer. Eventually it'll collide with us. And if we, instead of going right at the star Merak in Andromeda, if we go left, then we get to M33, the famous Triangulum galaxy, in Triangulum, magnitude 5.7, good deal fainter than M31. I've always found it much more hard to see through binoculars or certainly with the naked eye. But it's further away than M31, perhaps 2.7 million light years away. And I mentioned earlier that Jupiter was due south of the star Hammel. Well, if you find the star Hammel and find Merak in the middle of Andromeda, then M33 is more or less between them, halfway between them. Now, this is looking east in the mid-evening, and we have again, I can't use the lasers, but Orion is is climbing above the horizon, was climbed above the horizon between the east and the southeast, and it lies middle of this image. This chart is Capella in Auriga, and above that is Perseus and the interesting star Algol. Now, Algol forms an equilateral triangle. If you look at it between Algol and Jupiter and Aldebaran, there's currently a nice, fairly precise equilateral triangle in the sky to pick them out. Algol, the name is Arabic for demon's head, or the ghoul, and it represents the head of the Gorgon, one of the Gorgon sisters, Medusa, in Greek mythology. She had snakes for hair and turned anyone who looked at her into stone. Medusa was slain by Perseus on his way to rescuing Andromeda from the sea monster Cetus. Algol is famous as a variable star, and it's in fact an eclipsing variable star. And this diagram illustrates what's happening. You have two stars. You've got an orange or a yellowish K2 spectral type star, and a brighter bluer B8 magnitude B8 spectral type star, which go around each other and partially eclipse each other. You know, when the bright star hides a bit of the fainter star, you get a small dip in brightness. But when the opposite occurs and the brighter star is hidden by the fainter star, then you get quite a substantial dip in brightness, which takes its brightness down from its normal magnitude, about magnitude 2.1 or so, only just a bit fainter than Polaris, the pole star. And it drops quite a factor down to about magnitude 3.4. Now, this eclipsing, the fact that it's an eclipsing binary was first recognized by the English amateur astronomer John Goodricke in 1783. Now, Goodricke was deaf and had previously been a pupil at a school for the deaf here in Edinburgh. Algol is a multiple star. In fact, there's more than just two stars there. There's a third star which is not involved in the eclipses, that orbits the other two every 22 months. You can watch these eclipses of Algol with the naked eye, quite easy to follow. if the process takes a little bit of time, a few hours to fade and a few hours to come back, and these are the times of the minima of algal during this month, morning of December 15th, and then the evenings of December 17, 20, and 23, you should see the star fade down to a minimum at these times and then slowly recover. It takes a few hours for these for the brightness to change from bright to faint and vice versa. The view to the east also gives me a chance to mention the best meteor shower of the month, indeed the best of the year. The Geminids diverge from a point close to the star Castor in Gemini, and they diverge from that point. They appear in all parts of the sky. You don't just need to be looking in the east to see them, and of course Gemini is rising, passing high in the south in the early morning. You don't need to be facing the radiant to see them. You can look anywhere, but the tracks of the meteors themselves will diverge from the direction of the radiant. Details for the shower, well, it's active, said to be active from December the 4th to the 17th, but the peak this year is somewhere around the evening of December the 14th. I've got 19 hours GMT on December the 14th. In fact, the Geminis have a fairly flat peak, so it doesn't really change in brightness, in numbers rather, for the best part of a day. The meteor speed is 35 kilometers per second, and it says the zenithal hourly rate is 100 plus. Now that's the rate you would have, a number of meters you might see if you had a clear sky and the radiant was high in the sky, and the sky was dark, there was no moon. In fact, there was no moonlight interference this year, which makes this a good year to look at for Geminids. Now, the Geminids derive from an asteroid, not a comet. Most meteor showers come from comets. They follow comets. They follow the orbits of comets because they've been shed from the comet's nucleus. The Geminids are thought to derive from the asteroid Phaethon. Now, I did check before I came here how to pronounce that. I've always used to call it Phaethon, but Phaethon seems to be one pronunciation, but you can look elsewhere on the internet and you get it called Phaethon. So there's a difference between the pronunciation. You take your pick. Anyhow, the asteroid is 6 kilometers wide, and it passes closer to the sun, 21 million kilometers, than any other known asteroid. And until this year, it was thought that Geminid meteoroids were splinters of rock that had been broken from the asteroid by the heat stress of its close approaches to the sun. It's sometimes been called a rock comet. But data reported recently by the Parker Solar Probe, however, suggests that the particles in the stream were released in a single burst, perhaps as a result of Phaethon colliding with some other object. So that's something to watch for, certainly on the night of December 14th, 15th, but a night or two earlier than that or later will still give you reasonable meteor rates. This is our view looking south at midnight and mid-month. There you can see, I can't put it out, but Gemini is much higher in the southeast by this time, as Capella is almost overhead, above Orion. And if you take the belt of Orion, well, you can stretch it down towards Sirius, or stretch it up in the opposite direction, above and to the right, towards Aldebaran in Taurus, and then onwards towards the Pleiades. You can see over on the right, on the left-hand side, the sickle of Leo coming up, and I'll come back to that because there's something in Leo that's of interest too. But let's focus on this particularly dusty region of the Milky Way, and they're lighting up the dust as they're passing through. In fact, you can see in this image galaxy. Just above that, marked by a green square on the image there, but I'll highlight it if I can. There is M78, which is another image, another object which we've imaged with the ASERO, and this is Ian Smith's version of the data. M78 is Casper the Friendly Ghost Nebula. I'm not sure where that name's come from, but anyway, it's a reflection nebula. Another part of the Orion molecular cloud, the giant cloud of gas and dust It's being lit up in this instance by two young giant stars, and infrared observations show that there is a cluster of stars with many young ones involved as well, and probably new ones being formed. And it's pretty well the same distance as the Orion Nebula, about 1,350 light years away. There is one other object I want to highlight in this region, and we'd be hard-pressed to see it, even using the ASERO. Some of us saw it with the naked eye back in 1986. I'm talking, of course, about Comet Halley, which is down there in the lower left part of our chart, to the south of Cancer in the constellation of Hydra, the Hydra, the snake, whatever it is, Hydra. The comet is reaching its furthest point from the Sun on December the 9th, and it's moving in an orbit which takes 75.9 years to go around the Sun. And it's reaching aphelion, its furthest point, at 1 o'clock in the morning on December the 9th. That's predictions from the JPL. Magnitude 29.1 expected, and it's going to be 34.5 billion kilometers from the Earth, and a little bit further than that from the Sun. Back in 2003, it was observed, and that was the last observation of it that I know of, at magnitude 28.2, and it was photographed by European Southern Observatory in March 2003. And there you see it, right in the middle. Can you see a smudge? I'll highlight it just there. And that was Comet Halley in March 2003. And as I said in the Scotsman yesterday, I suspect there'll be several observatories trying to capture it at its furthest from the Sun around about now. Now I have to mention Betelgeuse, the red supergiant star in Orion's shoulder. There it is. Its brightness changes have been of interest over recent years because it dipped quite severely in brightness to fainter than we've ever seen it before, and there was speculation that it was going to explode as a supernova. In fact, it's recovered. It's now about magnitude 0.5, and it's near the top of its brightness variation range. It always varies. It's a pulsating red supergiant. This month, though, it could disappear entirely, but only for a few seconds, and only if you're in the right spot to see it. What is happening is an occultation on the morning of December the 12th, and it's being occulted by a 14th magnitude asteroid called Leona, a potato-shaped object measuring about 80 by 55 kilometers. And this was an observation taken on November the 19th of the asteroid moving against the star field. Now on the December the 12th, it'll pass directly over Betelgeuse. Leona is 45 milliarc seconds across, apparently, if you in apparent diameter, which is not very dissimilar to the apparent diameter of Betelgeuse. So we don't know whether the occultation will be total or partial, but we do know pretty precisely where the track is, and the track's going to pass through across southern Europe. It moves westwards, shown by the arrow on this image, on this illustration, and that's in the small hours of December the 12th. But it passes through Spain, but not over our observatory there. So I doubt if there'll be any, could be any observations of it there. Anyway, that's something that's happening of interest this month when one of the brighter stars in the sky, Betelgeuse, may disappear just for a few seconds as it's occulted by Leona. This is looking southeast at 7 o'clock in the middle of the month, December 16th, 7 o'clock in the morning before sunrise. Venus is still brilliant down there, but it's getting lower each morning. It's chasing the sun southwards in the sky, and this yellow line shows its motion against the stars over the course of the month. On the 16th, morning of the 16th, it's magnitude minus 4.1, brightest object in the sky apart from the moon, and 15 arc seconds in diameter, and 73% illuminated, but it's getting further away, and the phase, the illumination is increasing. It's becoming more gibbous than it was. So it's getting smaller, and at 15 arc seconds, you'll need a telescope to see the phase. It'll be to the left of the waning crescent moon on the 9th of December, and the following morning on the 10th, it'll be above the moon. The moon at that time will be quite impressively Earth-lit. Now, another object I want to mention, and I've already mentioned Leo, another object I want to mention is passing through Leo, and Leo is up here at the top right side of this chart for the morning sky. And there it is in close-up. The object in question is another comet, comet 62P Chuchinshan. I'm not going to try and pronounce that again. Right. This is the path of the comet through to the middle of January. Again, its positions plotted every 5 days and positions labelled every 10 days. This comet is smaller and closer to us than Comet Pons-Brooks, Pons-Brooks that I mentioned earlier. Currently, it's around magnitude 9 or 10, and it should be a little brighter than that by the time it reaches perihelion on Christmas Day. It's then 189 million kilometers from the sun, but only 80 million kilometers from the Earth, and it'll be just below the star Chertan in the hindquarters of Leo. Douglas Heggie posted an image he's taken of the comet on Tuesday, showing a typical comet appearance of a greenish coma around the nucleus. So that's the second brightest comet this month. I postponed, at the start of this talk, I postponed my traditional look northwards in the sky at the mid-evening time. But here it is. The plough is below and to the right of Polaris, the pole star, is starting its climb up into the northeastern sky as the W of Cassiopeia sails overhead at the top of the diagram. Castor and Pollux are in the eastern sky, with Castor lying above Pollux. And of course, Castor is where we see the radiant for the Geminids. And you can just see on the right-hand side the Milky Way and the summer triangle sinking down into the northwest. Now, December's second main meteor shower occurs later in the month, and it's called the Ursids, and it has a radiant near this position, which at this time lies directly below Polaris in the north. The Ursids are the sky, but they diverge, their tracks diverge from the radiant, and there are the details for the shower. It's active between the 17th and 26th. The peak is expected in the morning of December the 23rd, about 4 o'clock. And the meteor speed is 33 kilometers per second, which I think is rather similar to that for the Geminids. But the predicted zenithal hourly rate is only 10, and we've also got to contend with moonlight around that date. So it's not brilliant, but I say it's been under-observed. I'm not sure how keen you are on going out to observe meteors in the small hours of a day, just 2 days before Christmas, but anyways, there. I've also got to mention the Quadrantes meteor shower, which peaks in early January. I know this is a December talk, but it peaks in early January, but before our next meeting. Its radiant point is trailing the handle of the plough. In the evening sky, it's rather low down in the north, but it does climb higher as the night goes on. Climbs higher into the northeast. If we wind the clock forward to 4 o'clock in the morning of January the 4th, the radiant is there. It's 50 degrees high in the eastern sky for Edinburgh. But as you can see, we have bright moonlight in the southeast. So even though it's the peak of the shower at that time, sorry, I need to show you the meteors and the details. The shower is peaking at 9 o'clock in the morning, but so on January the 4th, but so it'll be best before dawn. But we'll have some moonlight interference. Now, the rates are quite high, 80 plus meteors per second. and it's got medium speed meteors, but the peak of the shower is fairly brief, only a few hours long. You can see shower meteors between December 28th and January the 12th, but most of the activity of the shower occurs in just a few hours around January the 4th at 9 o'clock in the morning, by which time it'll be dark, at which time it'll be daylight. The meteors are possibly related to another asteroid, one discovered in 2003, and that in turn may be related to a comet seen about 500 years ago. The shower can produce some bright, spectacular meteors. Back in the 1960s, I observed a quadrantid fireball of around magnitude -8 during a meteor observing expedition to the Isle of Man. That I'll always remember. I'm getting near the end now. I thought I'd finish with a nice astrophotograph, again from ASERO, this one processed by Ian Smith, of the Seagull Nebula, and I think it makes a fairly good bird-like impression. The Seagull Nebula IC 2177, it lies near the border between Canis Major and Monoceros. It's about 8 degrees above and to the left of Sirius, the brightest star in our night sky. Again, this was taken by ASERO, and the Seagull is a combination of an emission nebula and a reflection nebula. So it's not unlike the Orion Nebula, but it's further away. It's nearly 3 times further away than the Orion Nebula. It lies at 3,650 light years. And that's all I've got for you this month.
Thank you, Alan. That was really good. That's it for this evening. Thank you to Richard, and thank you to Alan for those really excellent talks. Our next meeting is online on the 15th of December, and that's about the science of Palomar Observatory. It's tea and coffee, so hang around afterwards, have a chat, and we'll see you later on in the month. Okay, good night. Thank you.